Epigenetics defines how genes and outside factors make us who we are. What our grandmother did to take care of herself may affect you. Epigeneticist Joyce Ohm is here to tell us how. Thanks for coming on the show today.
Thank you for having me.
Well, I tried to sum up what epigenetics was, but what do you tell people when they say, what do you do?
Well, the best explanation we have for the field of epigenetics is actually if you look in a mirror. So the DNA in all of your cells is exactly the same, and yet we're able to develop from a single fertilized egg into the person that you see in front of you. And epigenetics is the main tool by which cells do that. It's how we package away that DNA. It's how we decide what genes to turn on and what genes to turn off that we might need as we go through life. Okay, and how did you fall into this? Were you, as a young child, I want to study epigenetics? I don't think anybody knew what epigenetics was way back then. I'm a cancer biologist by training, and that's what I've worked on for the last 15 years or so, and actually stumbled into the field about a decade ago at a scientific meeting and sat down and thought, what's this epigenetics thing about? And ended up fascinated. I spent 2 whole days listening to some of the experts in the field explain it and headed off to do a postdoctoral fellowship with one of those experts. Well, what was the draw for you? What was the thing that got you excited about it? Well, there's so many diseases that we know have familial ties, but we can't explain them with just genetics. Obesity, cancer, neurodegenerative diseases like Alzheimer's disease. We know these run in families. We know diabetes runs in families, but we don't have genes that can completely explain those away. There has to be something in the environment that is playing an important role in whether those diseases are actually come to fruition or not. Okay, and you research this. How do you research it? We use a lot of very standard molecular biology and biochemistry techniques. We do everything from next generation sequencing and actually sequencing hundreds and hundreds of thousands of base pairs of DNA. We have large-scale array methodologies where we can look at 500,000 different modifications at a time. That sounds difficult. Is it hard, like in terms of how research goes, is it hard to research this? Well, fortunately, the technologies have evolved along with the science behind it. And in fact, there's fascinating questions. People have been waiting for technologies to appear that we can actually study them. And we're really getting to that exciting point. The field of epigenetics has exploded in the last 5 to 7 years, and we'll see even more of that. Sure. Well, and you mentioned how diseases play a role with genetics, of course. But what about in this area specifically? Give us an example of how a disease is included in this. So I'll give you one of the classic ones, and it's based on a long-term study that was underway in Europe, where they studied, this goes back to that grandmother question, they studied women whether they were pregnant during times of feast or famine, and actually showed that their grandchildren had an altered risk for developing diabetes, depending on whether it was a time of plenty or a time of want. So women, when they were pregnant during times of famine, developed methodologies in their genes to actually store nutrition and really hold on to those calories. Well, we don't need those anymore. We can walk into a supermarket and buy anything we want now. But those mechanisms have evolved over time for us to really be able to hold on to that fat and hold on to that energy. And now you're seeing that in subsequent generations. So our genes, of course, they take a lot longer to change than a lifetime, I guess. They do. And epigenetic traits in particular can be passed down from cell to cell and from generation to generation. And so those lifestyle choices we make, things like smoking and drinking and exercise and diet, may actually play a role in future generations' health. And that's what you're finding then, that the way we're treating our bodies right now will affect? I think we're absolutely seeing those things in animal studies for sure, and some of the first human studies are coming along. We've got great researchers here at UND looking at a variety of different conditions, but one of those is looking at preeclampsia and the role that epigenetics plays during in utero development. And they've shown that actually offspring can have an increased risk of cardiovascular disease and heart disease later on in life, and those risk factors are being passed down from generation to generation. So as you're going through this research, finding these things, are you saying in your head, boy, I should do certain things to make it better in the future? People ask that a lot. I think we have learned that we probably do have some responsibility to our epigenome. You're born with the genes that you're born with, but we do have some responsibility for the rest. The flip side of that is most epigenetic changes are reversible. So when we have changes like these that are affecting disease state, these are things we can actually respond to. We have drugs and therapies in the clinic now, in particular in cancer, that can actually reverse some of these changes. Sure. So that's encouraging that this is something we actually have a little bit more control over than our genome. Well, one last question for you. What do you focus on in particular with your team here at the University of North Dakota? So I'm a cancer biologist by training, and what we're studying is environmental agents, things like pesticides, herbicides, endocrine disruptor chemicals that we find in the environment, and looking at changes in your epigenome during development.
Well, it's a fascinating topic. Good luck on your research.
Thank you so much.
Thanks for coming today.